the accumulation of mutations in these separate lineages over time. So according to Zucker, Candle, and Pauling, when you make mistakes, they stay in the molecule, and subsequent mistakes stay in the molecule. So the longer it's been since these molecules were one molecule, after all, they're all copies of this ancestral molecule, the longer it's been since they were that single molecule, the more different these molecules would have become due to the accumulation of these random mutations. Okay? Now, Zucker, Candle, and Pauling say, well, if you can, you could calculate the likelihood that a particular mutation occurs, right? These mutations occur during DNA replication at certain frequencies, certain probabilities that these mutations will accumulate in the molecule. And if you know what that probability is, or that rate of mutation, then you're halfway there. The other half of the process is you have to know how long it takes to take this DNA molecule and move it to the next generation. Well, we've done this concept before when we did life tables. This is in fact what's called the mean generation time. The mean generation time is the length of time it takes to produce those offspring. Remember we talked about the average amount of time it takes the female to produce offspring, those however many offspring are sub zero over her lifetime. Well, that's the mean generation time. If we know the mean generation time and we know the rate of mutations, what we could do is look at two DNA molecules today, and based on the number of mutations and the length of time between generations, we could determine how long it's been since these two molecules go back to one molecule. Okay? So this concept here is called the molecular clock. And in the molecular clock, what we're going to do is use the number of mutations, right, within the sequences that makes them different, and use that to figure out time since divergence. And that should look like this. If the rate of mutations occurring is constant through evolutionary time, and if the mean generation time is constant, then the number of mutations different tells us the time since divergence. So we'll use this molecular clock technique to figure out how long it's been since species go back to that original individual. Differences among these two individuals with respect to their sequences tells us how long it's been since that common ancestor for both walked the planet. Okay? So we're going to use this idea of phylogenetic trees, phylogenies, and the molecular clock to try to figure out something about HIV. All right, so in this phylogenetic tree, we have a number of different viruses that we're going to keep track of. The viruses will fall into two main groups. The human immunodeficiency virus labeled as HIV, and another one called the simian immunodeficiency virus, which is SIV. Okay? Now, we have HIV from humans, obviously. It's a human immunodeficiency virus. And we have SIVs from a bunch of different types of non-human primates. We have an SIV from a Sykes monkey, a stump-tailed macaque, a pseudimangami, an African green monkey, a mandrill, Loest monkey, sun-tailed monkey, and chimpanzee. So we have a number of these. What we're going to do is take the RNA molecules from HIV and sequence them. instead of ACs, Ts, and Gs, we're going to get ACs, Us, and Gs. But I should have a particular sequence of letters that represents the RNA molecules found within the genome of HIV and from SIV. Let's take all of those sequences and have the computer figure out which sequences are more similar to each other with respect to their sequence, the identity and order of the letters. Okay? All right, now I've got that sequence. I can build a phylogenetic tree, and I can put a timeline on it. So let's build the phylogenetic tree first. Notice that according to the phylogenetic tree, at this point in time where the red arrow is, this is the ancestor of all of the IVs, the immunodeficiency viruses, from which the human and all of the simian immunodeficiency viruses descended. Okay? We get a bifurcation, right, a diversification that leads us to this clade and this entire clade here. That would be the common ancestor there. This ancestor split into two more clades. So for usefulness, I'd say we have pretty much 3 clades there, although you could make an argument that there are 4, maybe the red, this blue, and that green. But if we keep the point here, then we have a blue, red, and a green clade. There are 3 clades there, right? 3 ancestors that gave rise to the 3 different groupings of the immunodeficiency virus. The first group, the green group, has humans in it and the pseudomangabe and the stump-tailed macaque. The blue group doesn't have any humans in it. It's all simian immunodeficiency viruses, but for different simians. And then here at the top in red, there is human and chimpanzee. Now, that leads to a question here that could be answered using this phylogenetic tree. We could ask, well, what is the ancestor of the human immunodeficiency virus? And the answer would be, well, it's a simian immunodeficiency virus that diversified through a group of primates, including humans. And then we could ask ourselves, well, how did the simian immunodeficiency virus end up in humans? Well, it appears that the simian immunodeficiency virus somehow crossed over to humans here based on the phylogenetic tree. And then we could say, okay, well, how many times did that happen, and what simian immunodeficiency virus is most similar to the modern day HIV? Okay? So here, based on this phylogenetic tree, it looks like there are 2 different types of HIV. There's an HIV here that's more similar to pseudomangabe and stump-tailed macaque. That's what they're calling HIV-2. And then there's one in red that's similar to the chimp's SIV, which they're referring to as HIV-1. The blue here is the main simian immunodeficiency virus. So it looks like one of the HIV source was either of these 2 species, whereas HIV-1 source is the chimpanzee. So far, so good. Then we could say, well, how many times has this virus kind of jumped species? Well, down here, it looks like it's happened at least twice based on this phylogenetic tree. There's a bifurcation here between the SIV and the HIV, and here there's another bifurcation between an SIV and an HIV. So within this group, this phylogenetic tree says, well, HIV-2 crossed at least twice. And up here for the red HIV, it looks like it's crossed over twice. There's one here, and then the second crossing over occurs there. Now, the data that you have that you use to build the phylogenetic tree, the more data you have, the more information you have. So what we're going to do is take and add more individuals here to the red part of the tree. So this star represents that star. This is this part of the tree with more data. They've added more individuals, and look at this. Now they've got more clades. There's a clade here called HIV1 group O. There's a clade here that's group N, and this clade, which is group M. So that would suggest that the simian SIV, right, crossed over to humans multiple times. Not once, but multiple times. And we've seen this kind of over and over again where I've showed you in class, right, talking about species coming in contact with other species, and then that allowed for the transmission of virus or other pathogen between species, and that's the origin of some of these emerging diseases. And we're all home now because of the coronavirus. Well, that one appears to have originated, what, in a bat that they had at a wet market. This one originated with simians, spread through different species of non-human primates, and then looks like it moved through species barrier from these non-human primates into the human population multiple times. Okay? All sorts of folklore around how this all happened, but we might be able to answer it by actually sticking a timeline on this. So we're going to use that molecular clock approach, look at differences in these sequences. We know the rate at which mutations are introduced into the HIV RNA molecule and the SIV RNAs, and we know the mean generation time of all these species. So based on that, the virus jumps to human populations in the 1930s. In Africa, because that's where these non-human primates exist. But what's interesting is the current thinking is back in the 1930s, the colonial powers had invaded Africa and had begun to take over parts of it. France was there, the Netherlands was there, Germany was there, and England was there. Like the big English colony would be South Africa. And then as part of the colonization of Africa, the European powers were after resources, things like gold, silver, diamonds. And if they could get access to those, it was worth a lot of money. So as part of getting the resources out of Africa and back to Europe, they built lots and lots of roads. And the way you build roads is you start at one place, you hire a group of guys to build the road. They're going to camp out right here, right, in little tents. They're not going to go back home every day because this could be hundreds of miles, so they camp out. And as part of camping out, you need food, and you need chefs. So you had the cook who provided food for everybody, and the thinking is at some point the chefs began resorting to non-human primates. Maybe they couldn't get anything else. I don't know. Maybe they're a delicacy. I don't know much about that. But in any event, the thinking is that that's how it first moves into the human population, is during the building of roads in the 1930s in parts of Africa to transport all these goods and materials out of the continent. Okay? All right. Okay. Now, in terms of the virus, we do know that there are people on the planet that seem to be naturally immune to HIV. And when their biochemistry was analyzed, it turns out that these individuals have the same old CD4 receptor that we expect. That's the receptor, right, that's used by HIV to recognize the CD4 cells. Then, if you remember, you bind to the CD4 receptor, And then the next protein that you have to bind to is called a Ccr5 receptor. You bind to the Ccr receptor, then you bind to the fusion domain, and the binding to all three of those brings the virus close enough to the cell to fuse and dump its content into the cytosol. Well, this Ccr5 receptor is kind of interesting because there are a number of alleles in the population for the Ccr5 receptor. One of those alleles is called the Ccr5 delta 32. Delta here means deletion, and the 32 refers to 32 nucleotides. So there are people in the population that have a Ccr5 delta 32 gene, but within that gene there's a deletion of 32 nucleotides. That means the protein that's produced is a little shorter and is missing some amino acids in the middle, probably about 10 of them. Does that make sense? Other individuals in the population have an allele that's much more common. We'll call this the Ccr5 wild type allele. The language wild type just means common. I prefer not to use the word normal whenever we talk about genetics, things like alleles, because I don't know what normal is. You find a normal human, show me, I'd love to meet him. I have yet to meet a normal human. And also, the other reason is because what's common in a population depends on the population. In populations close to the equator, what may be common is the allele that codes for brown eyes, whereas in Norway, it may be common, that may be the allele that codes for blue eyes. So this is very situation dependent, right? The wild type allele is the common one, and then this other one, the Ccr5 delta 32, is referred to as the mutant allele. A mutant allele, because the thinking is the common one is common because it's the typical sequence, and that sequence had to be modified to get the mutant. In this case, you have to delete 32 nucleotides to get that delta 32 allele. Okay? All right, the notation